These notes will be describing some of the information from Chapter 11, which is skeletal muscle. And the beginning part of Chapter 11 deals with the neuromuscular system and some of the basic structure of muscle. And I'm not going to do too much about that. You should review the beginning of Chapter 11. And so with this short video and some of the other short videos, I'll get into some structure, but then also muscle contraction, so that's important to know. And then a little bit about muscle training adaptations. And so actually, just as a review, we can remind ourselves from the beginning of our class the different energy systems. And we're using all of these with muscle contraction. So if we look at this picture, and I copied this actually from the web, so it's not out of our book, but that immediate energy system, which is phosphocreatine, that will make ATP over here on the right-hand side. Glycolysis, fast or anaerobic glycolysis, will make ATP over here. And the long-term aerobic energy system, either using a fatty acid or using pyruvates that we got from glycolysis, will make ATPs. And then these ATPs are required for muscle contraction and relaxation. And the enzyme that breaks down that ATP to allow the actinomycins to get together is an ATPase. And in fact, it's a myosin ATPase. It's located on the myosin protein that's found in skeletal muscle. So a really nice review of the different energy systems. And a lot of our body weight is muscle mass, and a lot of different muscles around the body. And in terms of function, yeah, of course, we are in kinesiology, we're doing human movement, so that's locomotion. Also, breathing, we don't think about that much, but luckily we don't think about it, but that's also muscle contraction is also required. And just to stand in one place for postural support, muscles need to be contracting for that support. And also heat production during cold stress. And maybe we don't think about this as much as a major function of skeletal muscle. But actually, if you remember from earlier, we talked about some uncoupling proteins and brown fat in mitochondria. So this is non-shivering thermogenesis. If we would shiver, right, that's little muscle contractions, and that's producing heat energy to keep us warm. So a nice review of some energy systems and some things we've talked about earlier, and some basic review, probably from an anatomy and physiology class, of the structure of muscle. And our current textbook has a great picture in figure 1115. This is a slightly different version of that same picture. I think I copied it maybe from an older version of the textbook. But I always think of muscles as tubes within tubes. So here's a whole bunch of different divisions of, either a gross muscle belly, like quadriceps or something. And then they have all these different divisions until we finally get down to a single muscle fiber or muscle cell. And just to review, make sure you look at figure 1115. It's actually a nice description of some of the structure of muscle. Also, be able to understand the difference as you're looking at a muscle fiber or muscle cell or some of the gross structure. Understand the difference between cross-sectional and longitudinal view. So here across the top, this was cut and sliced that way across the top, and that would be a cross-sectional view. If we look at it down the side of this is a longitudinal view. And muscle cells are called muscle fibers. They're long sort of fiber like if it's a piece of clothing fiber, so that's where its name came from. And within this muscle cell are these myofibrils, so that's either even further divisions of muscle. And then there are also different light and dark bands. So make sure you sort of remember what those light and dark bands are. That would probably be from an anatomy and physiology class, but an A band, I band, and actually in our current version of a textbook, that's page 347. And what else is fun in here? So the sarcoplasmic reticulum. A reticulum is a network of tubes. So you can sort of see these networks of tubes in there. And those tubes contain calcium. So inside the sarcoplasmic reticulum is calcium. We've talked a lot about mitochondria. Here's a mitochondria surrounding this, or inside this myofibril. Here's a transverse tubule that actually goes from the outside of this cell deep into the inside of this cell. So it actually transverses, it goes through this cell. Up at the top at this cross-sectional view, you can sort of see it, but here's dots that would represent actin-myosin proteins, or the thin and thick filaments. And here they are along here as a longitudinal view. So sort of a nice little structure of this muscle fiber or muscle cell. And here's another view of it from a different picture. Here would be a muscle cell, and they pulled out a myofibril. And so this would be a longitudinal view, and here it's been sliced, so that would be a cross-sectional view. There's filaments of actin. The thicker ones would be filaments of myosin. And here are the different bands, like H zone and the Z line, A bands, I bands. Just make sure you remember that stuff. The functional unit of a muscle cell in the myofibril is this sarcomere. And so sarcomeres are in series, so there's a bunch of sarcomeres in a row. And here are the Z lines that attach these sarcomeres. So another nice picture out of our book of the structure of, more structure of muscle cells. And here's another figure, and it's just sort of drawn, but here are the different actin and myosins. Here's a sarcomere from Z line to Z line, and here are these different bands within this sarcomere. And what we want to get to is muscle contraction. So the key to the sliding filament model, and it's still considered a model and hypothesis, is the Z line to Z line distances are going to decrease. So a sarcomere distance is going to decrease. And not because the actin and myosin proteins change length, but because there's a greater overlap. So there's a greater overlap of thin and thick filaments, or a greater overlap of the actin protein with the myosin protein. So the Z line to Z line distance is going to decrease. And here's another nice picture out of our textbook. This is actually figure 1121. And obviously this top panel is a resting state, and the sarcomere length is greater. And with greater amounts of overlap, here's a contracted state. And so you can see there's greater overlap of the actin proteins with the thicker myosin proteins that are found in the middle. And so here there's complete overlap. So this is obviously a contracted state, and the sarcomere, the distance from this Z line to this Z line, is shorter. So in our next notes and our next videos, we'll review the current idea, the current hypothesis of how muscle contraction occurs.